Hello, everyone. Welcome back to today's EuroVA workshop. To be more precisely, the second paper session, which is starting right now. My name is Jürgen Bernard. I'm at UBC. I'm very happy to chair this session. I hope that you can hear me well. And greetings from Vancouver, by the way, which is still quite dark. I'm hearing something from my... Am I on air? I will stop talking. Okay, wonderful, thank you. In this session, we have 5 wonderful papers that we'll present all across the lines of high-dimensional data and time-oriented data about quality measures, explainers for dimensionality reductions, progressive analytics, time series analysis, scaragnostics variants for time series, and some event sequences related material. So a lot of interesting VA stuff. I was asked to remind the audience that you can give feedback and for particularly questions in YouTube as well as on the EuroVA channel at Discord. Also, please use emojis to raise questions, and then I will present these questions to the speakers. Of course, you can also pose questions to me as far as I will be able to get these. I would like to proceed and come to the first speaker that we have today, which is Sarah Johansson-Fernstad. She's lecturer in visualization at Newcastle University. She's active in our community for over 10 years, as far as I know. Her interest is in missing data visualization, biological visualization, genomics, which will also be a part of the paper today. We all love her works about quantifying of mixed data in 2008 and about quality metrics, which is actually quite suited to introduce her today's topic, which is quality measures to guide visual analysis of high-dimensional genomics data.
High dimensionality is a challenge in data analysis, especially with biosciences, where genome sequencing techniques are able to generate data with thousands of dimensions. The use of quality metrics is a common approach in visual analytics to deal with high dimensionality, and this paper presents a set of quality metrics of relevance for visual analysis of high-dimensional genomics data, which is based on a survey with domain experts. So genome sequencing is increasingly common in many domains. These include medicine and health, environment, personal and home care, and agriculture, to mention a few. Looking at the basic data structure of this data, which applies to most types of omics data. First of all, the data comes from samples, which from data point of view corresponds to data items. Often they are structured in sample categories such as test and control that are relevant for analysis. So DNA is extracted from these samples and it's analyzed to define biological entities. And exactly what these biological entities is depend on the type of omics data. If you look at microbiomics data, for instance, we often talk about operational taxonomic units or OTUs or species. So these biological entities correspond to data dimensions. The output of this is a table of counts, which basically are the abundance profiles for the samples. And these tables are often highly skewed with a small number of biological entities that are detected in high counts in many samples, and at the same time a large number of entities that are detected at very low counts and in only a small number of samples, and this affects analysis and which analysis methods that can be used. So in this presentation and in the paper and supplemental material, I use 2 different example datasets. The first one is data from a study of the gut microbiome of preterm infants, which is in the context a fairly small dataset. And the sample classification that we look at there is birth mode, where we have cesarean and vaginal birth. The other dataset is considerably bigger. It includes samples from different oceanic areas. And the samples here are categorized by layer of origin, with 3 different levels, where the surface is the top level and the mesopelagic is the deepest level in the oceans that it's sampled. So, looking at the use of quality metrics in high-dimensional data, quality metrics can be defined as calculated metrics that capture data properties that are useful or that are interesting for the extraction of meaningful information about data. And in the context of high-dimensional data, we want to use them mainly to select subsets of dimensions or of biological entities that are particularly interesting for further analysis. And the question is then, what is interesting? And we argue that what is interesting is, at least to some extent, domain dependent. There is a difference of what is interesting in biology and looking at genomics data compared to when looking at finance data, for example. So what we want to do is to identify patterns in genomics data that are particularly interesting and relevant for further analysis, to use this as a basis for defining quality metrics. And we define these through a survey with domain experts. So what we first did was to define a number of data patterns of interest based on our previous work and refine these basic patterns in discussion with 2 microbiologists that we worked together with. And then we conducted a survey where we have participants ranking the interestingness of these patterns using a Likert scale where 1 corresponds to not interesting and 5 corresponds to very interesting. And also gave the opportunity for the participants to add other patterns through free text options. So looking at the survey results, the survey was carried out by 20 scientists with expertise in bioinformatics, microbiology, and other biology, and they were from a range of different application domains. So looking at the results, we looked at the ones that were defined as interesting or very interesting by the participants, looking at the percentage of participants that replied that these patterns were interesting or very interesting. And based on this, we defined 5 different categories of patterns of interest. So the first one is the abundance and prevalence of biological entities, looking at the individual entity values. Second one being the difference in abundance or the difference in prevalence between different sample groups. Third, looking at multivariate entity relationships, which is the correlation and the similarity between different biological entities. Fourth one is related to the biological hierarchical taxonomy. So as part of the analysis, entities are classified using this hierarchical taxonomy, and the output also provides a certainty value in terms of classification, which is of interest for analysis. However, since this certainty value is already given through the initial classification, we didn't see this as something that's particularly valuable in terms of defining this as further as a quality metric. And the fifth and last category was the relationship between biological entities and individual samples, and the relationship between biological entities and groups of samples. And this is a type of relationship that we found is a bit too complex to describe as a single quality metric, so as a simple quality metric, and something that's better investigated and understood through interactive exploration. So based on this, we chose to focus on the first 3 categories, and I will now show you some examples of how these can be used in visual analytics to select interesting subsets of biological entities for further investigation using the 2 different example datasets. So starting with the first type, the first category, we have the individual entity values, and they were looking at abundance, which is the total count that is detected in an entity in all samples, and the prevalence, that is the relative number of samples that an entity has been detected in, so it's a percentage value. So in the plots here, the top one is the 10 biological entities that were highest ranked for abundance, and the bottom one is the 10 highest ranked for prevalence. And the axis in the parallel coordinates corresponds to biological entities, and the polylines to samples. And here we're using the Tara Oceans dataset, and we can see that the top plot has considerably higher maximum detection values, indicating that this is something that has been detected in high counts. We're looking at thousands as a maximum value, whereas the bottom one are entities that are detected in all samples at some level, because the minimum value is above 0, but they haven't necessarily been detected in high counts. So this can first of all help to identify the most common biological entities occurring in many samples and at high levels. But it's also interesting to combine these two different metrics to find outliers, which could be, for instance, entities that have a low prevalence, so they're only detected in a small number of samples, but they have a high abundance, so they're detected in high counts. The next group of metrics is the sample group differences. So differences between sample groups are often core of studies, so comparing healthy and unhealthy participants in a study, for instance. And we start with the prevalence difference between groups. So the prevalence can be described as a percentage of samples that an entity is detected in. And the prevalence difference can then be defined as the difference in prevalence between groups in an entity. So we're looking at the average difference in prevalence between the sample groups. Here we look at the data from the preterm gut microbiome, and we have the 10 entities where the prevalence between entities, the difference in prevalence between entities is high. And we can, for instance, see in the leftmost axis where we have only prevalence, only the blue samples are prevalent in this entity, whereas they're not prevalent at all in the red samples. And this can help identify biological entities that may be involved in driving differences between the birth modes. The second part here is the abundance, and using average difference doesn't work as well for abundance as it does for prevalence, because there can be a lot of variation in abundance between samples, whereas prevalence is something that either it is prevalent or it's not prevalent, so we can get a percentage value, but the variation is considerably higher when it comes to the abundance or the counts. And we can compare this to clustering, where we have cluster centroids that possibly are far away from each other, but we still have clusters that overlap. And here we see an example of the 10 entities that have the highest average difference in abundance in the preterm data. And we can see quite clearly that the blue and the red samples are very mixed up, so it's not a good metric for identifying difference between sample groups. So what we suggest is to instead use a cluster separation approach, where we're treating sample groups as clusters. And the aim then is to identify entities where the separation between sample groups is high using a cluster separation metric. In the example here, in the top plot where we're looking at cluster separation, we use the Davis-Bouldin index, but others could be used as well, such as the silhouette values. So what we see in the top plot here are the 10 entities where the sample groups are most separated using cluster separation. And we can see some clear examples of where the abundance difference is high between the groups. We have a couple of entities with considerably higher abundance in blue samples compared to the red ones, and the other way around for some other entities. So this again can help identify entities where the differences in these entities may be driven by the birth mode. So looking at the third group of quality metrics, we're looking at the similarity and correlation. And these measures provide a description of relationships between pairs of biological entities. And from biological perspective, they can indicate coexistence between entities and support identification of entities that potentially have a symbiotic or antibiotic interaction. And these are entities that tend to occur together in high numbers if they are symbiotic, and the opposite way around for antibiotic interaction. So there's a range of similarity and correlation measures that have been suggested for genomics analysis, and in this example that I'm going to show you now, we use Pearson correlation, but the paper lists a number of other options in terms of similarity and correlation. So as I mentioned previously, we often see a skewness in data when it comes to abundance, and there's a lot of entities with low or zero count for most samples. And these tend to have strong correlation mathematically, but they are not really interesting for finding coexistence patterns, so we suggest using a threshold to remove these low abundance or low prevalence entities. So a quick example here, we're looking at the preterm infant data again, and we see the 5 entity pairs with the strongest correlation in this data set. So it's the same data in the upper and lower plots, just showing them in using different visual representation. And first of all, what's visible is that there are not really any clear strong correlation patterns in the data. There is quite a lot of noise in the samples where there's lots of entities that are detected in low levels. However, we do see some patterns, such as, for instance, kind of negative correlation in the second plot and in the fourth plot. It's not a clear negative correlation, but we can see that there is at least no symbiotic relationship, since there are no samples that occur in high counts in both entities at the same time. And this is an example of a pattern that could be interesting to know a bit more about. So, looking into the future, genome sequence data will continue to be important and increase in terms of domain and in terms of size of the data. And visualization will become increasingly important as more domains mean more people with different backgrounds needing to understand this data. Quality metrics can be useful to help support analysis of this type of data, and future work includes to incorporate them into an interactive visual analytics system and to evaluate them to make sure that they are useful.
Thank you, Sarah, for that presentation. I would like to ask questions, but first we have a question from the audience, and I would like to start with Chatai, who is asking about your observations of transferability of these quality metrics regarding other application domains.
Okay, thank you for that question. So we haven't done any particular studies on transferring it to other data domains yet, though it is a good suggestion for future work, definitely. I would say that some of the metrics are definitely applicable to other domains as well. You could think of the abundance, for instance, as basically a numerical value, and as long as it's interesting to have either high or low values, it would be directly applicable to any other type of data as well. I think also the separation of sample groups in this case would be anything that's categorized. So thinking back to your paper in the previous session, there's quite a relevance of the work you're doing there. So that would also be applicable to data, numerical data, where you have categories that you're interested in, if they are well separated or not. The prevalence may not be as applicable to other types of data. I can't, at the top of my head, think of any data where the existence or non-existence in such a binary relationship is as important as it is in genomics data.
Sounds plausible. You already mentioned some aspects about data and data types, which was an interesting communication between Gennady and Chatai. I think there is nothing that much to add at this point. Or do you want to comment on that, Sarah, about data types in specific?
Data types in terms of numerical versus categorical, or yeah. So I think, again, some of it could be applicable to other types of data. The prevalence, I suppose, could potentially be seen as a categorical measure, so it could be applicable to categorical data as well. And again, the cluster separation approach could be applied to categorical data as well, but I think you would need to use different metrics, because obviously a class separation metric that's applied to numerical data is probably not that well working for categorical. I think temporal data was mentioned as well, and I haven't tested anything over temporal.
You also see the chat, because this would have been my next question. Sylvia asked, what about time? And this is different.
There would be definitely something that would be interesting to develop metrics for as well. Changes over time, absolutely. There was one of the participants in the study, I mentioned in the paper at least, that we did give them an option to have to add metrics that they would be interested in. And I think there was one or potentially two who mentioned time as something of interest, although it wasn't sort of mentioned across the participants.
Makes sense. Thank you, Sarah, once again for both presentation and answering questions. And I would now lead over to the second paper of this session, which is presented by Alexandru Talea today, who is a professor in computer science in Utrecht University in the Netherlands. Lots of visual analytics works in the last years about high dimensional data and dimensionality reduction, just like the work today. And the title is Attribute-Based Explanations of Multidimensional Projections. Looking forward to it.
My name is Alex Teller from Utrecht University in the Netherlands, and I will present you the paper Enhanced Attribute-Based Explanation of Multidimensional Projections. Take a high-dimensional dataset. Shown here is a table where each row is an observation having multiple attributes or dimensions. Projections map every sample in such a dataset to a 2D point so that the distances between points typically reflect as much as possible the similarity of the respective rows in the table. So a projection is simply a 2D scatter plot. But a raw scatter plot is not very useful. So how to explain the structures we see in such a scatter plot? There are many methods for explaining projections. We can color-code the points by the value of one attribute, but how to select this attribute? And is there a single one that can explain well all the points? Or we can visualize the errors in the projection, such as false neighbors or missing neighbors. However, this explains the projection accuracy rather than the projection itself. Or we can explain which variables map best to the spread of the points along the x, respectively y-axis of the scatter plot. But this assumes the projection behaves uniformly in a global sense. And finally, we can explain local neighborhoods or close points in the projection. This is a class of techniques that I'm going to focus on next. One example of such a technique is the work of Da Silva et al. Presented at EuroVA in 2015. Consider a simple 3D data set having points spread over the three faces of an axis-aligned cube. Consider two data points, x and x prime, in this data set. The contribution lambda xx prime superscript j of the jth dimension to the distance between these two data points can be computed simply as the distance along dimension j between these 2 points divided by the distance along all dimensions, that is in nd, between these 2 points. This can be extended to compute the contribution over an entire neighborhood of the jth dimension simply by averaging the point pair-wise contributions I described before. And finally, we can normalize this per neighborhood contribution so that it ranges with values between 0 and 1. Intuitively, if this contribution is low, then dimension j matters little to the interpoint distance, which means that dimension j explains well why points in the neighborhood are similar. Finally, we can color the projected points by the dimensions having overall low contribution over all the points. So, in our figure we see the three faces of the cube colored by the dimensions x, y, and z, which is indeed correct, because recall, these faces are orthogonal to these 3 dimensions. Finally, we can use brightness to show the confidence of the explanation, that is, how much the similarity of the points in the neighborhood is explained by the single color-coded dimension. So we see that points along or close to the edges of the cube are dark, since obviously in these neighborhoods similarity is due to 2 dimensions, not a single one. In our paper, we extend the work of Da Silva with 2 additional explanations. First, we consider a dimensionality explanation. That is, we want to show the user how high dimensional is the data at every place in the projection. For this, for each neighborhood, we compute the local dimensionality, delta, using principal component analysis. And we tried 2 flavors of this. Total variance defines local dimensionality as the minimal number of eigenvalues needed to capture a given user percentage of the data variance, say 80 to 90% in the neighborhood. And minimal variance is the number of eigenvalues larger than a user set minimal variance. We see how this works for the simple cube dataset. Over the faces, 2 dimensions are obviously sufficient to explain all the data variance. However, close to the edges, we need 3 dimensions for this. Next, we aim to explain correlations of the dimensions. That is, find which dimensions are strongest correlated around every point in the projection. For this, we compute a Pearson correlation of all dimension pairs over each neighborhood. Then we sort or rank dimension pairs in decreasing correlation strength over each neighborhood and then select the C most frequent ones over all the neighborhoods and color code them. Here, C is typically the size of a categorical color map, like 8 to 10 entries. Let's see how this works for the cube dataset. In the left image, we see the original attribute contribution of De Silva. The top face is red, meaning that X changes little here. The correlation view shows that the Y and Z dimensions are strongly correlated there. The YZ dimension pair is color-coded yellow. The same is true actually for the cube edge orthogonal to this face, which is also yellow, since Y and Z both change very little over this edge, while X changes a lot. For n dimensions, we have n squared divided by 2 possible correlations, but we can only color code about 8 to 10 of them. So how can we handle this when the number of dimensions is large? Or when we want to examine specific correlations. Take the left view, which shows the strongest and most frequent correlations over all neighborhoods. The dataset used here has about 1,000 samples of physical-chemical properties of different types of concrete. For this dataset, we have potentially 32 correlation pairs to show, but we only want to use 8 categorical colors. We address this using a matrix view. Rows and columns are dimensions, and the cells are dimension pairs. The colored cells here show the 8 dimension pairs of strongest correlations, which are color mapped in the projection. Blue cells show variable pairs, which are either weakly correlated or strongly correlated, but only over a few points. Still, if we want to examine a specific dimension pair, we can proceed as follows. For example, suppose that the user is interested in the fly ash C aggregate correlation. The user then can click in this cell, at which point the color allocated to the last of the top 8 variable pairs, that is light blue, used for water C aggregate, is removed from the color map and allocated to the user indicated fly ash C aggregate explanation. So if we look at the projection plot now, we see an outlier neighborhood, a small neighborhood colored light blue, which indicates that these 2 variables are indeed strongly correlated, but only over a small subset of the points. Let us now see how all the 3 explanations interplay to help understanding a projection. For this, we use a wine dataset having about 6,500 samples of wine types, each having 12 physical-chemical attributes measured for that particular wine. We start with a raw projection computed by the lamp technique. We can use any projection method if we want. We see a scatter plot which is pretty uninformative. Next, if we use the attribute contribution, the projection splits into roughly 4 zones. Each has wines whose similarity can be explained well by a single attribute. For example, in the red zone, all wines are similar, mainly because of their sugar percentages. Points close to the borders of the region are dark, meaning that we need more than one attribute to explain why wines are similar there. If you bring in the correlation view, we see that wines in the red area, in the attribute contribution view, having similar sugar percentages, also have fixed acidity strongly correlated with pH or with citric acid. So the correlation view refines the explanation of the contribution view. This also works conversely. If we look at the correlation view, we see a large purple area where wines correlate strongly in sugar versus density. The attribute contribution view refines this by splitting that area into wines similar due to alcohol, chlorides, or acidity. So the 2 views help or complement each other. Now, we found several local explanations for the wine dataset using the contribution and correlation views. Are those explanations sufficient? The dimensionality view helps answering this. The view shown left for 85% of explained variance shows that top left, the projection can be explained by roughly 3 to 5 dimensions. The combined correlation contribution view gives us an explanation in this area using 5 dimensions. Or in simple words, we have enough explanations here. We're happy here. However, in contrast, in the bottom right area, we see that we need 6 to 8 dimensions to explain the data structure. The combined correlation attribute contribution views only explain this area by 5 dimensions. So we just found out that we're missing additional explanations in this area. Concluding, we have proposed 2 new explanations of multidimensional projections based on dimension correlation and local dimensionality. Together with the original attribute contribution, we have now 3 tools which work jointly in explaining a projection. The proposed explanations are simpler to implement, fast to compute, and work for any projection method. However, when the number of dimensions in a projection is higher than a few tens, more work is needed to extend these explanatory mechanisms. Also, it is interesting to think about capturing and showing more complex patterns. Ideally, we'd like to go towards a skagnostics approach, that is, compute locally a wide set of explanations for a pattern library, for example, find the best ones locally, and then let users explore the entire set of explanations interactively. Thank you for your attention.
Thanks, Alex, so far for your presentation. Very interesting stuff and a very nice extension of the work that you did in 2015, I think. We have not received questions so far, so I will proceed with my first question, which regards dimensionality view. To spend a bit less time to explain this one, even I think it's very interesting. My question would be, we now have a means to sort of find out which the true or intrinsic dimensionality of data sets are. Can you tell us a bit more about your experiences with that technique, which is still new to us? What do we have to expect here? What's the benefit?
Okay, so putting it simply, it goes like this. The dimensionality view would tell you, roughly speaking, in a projection how far the 2D scatter plot there is from the number of high dimensions that you have in the initial data set. Or in other words, if you see, like in my slides before, red, in a local area in a projection, it tells you that the projection would have trouble in explaining a reasonable number of the dimensions in the original data set. So, putting it simply, it's an indicator telling you that it will be very hard to classify or to describe that area in the projection by just the few color-coded attributes that you've seen before. Conversely, if you see in a projection that there are areas that have, where the data, has a low dimensionality, then you'll know that with, the limited 3 to 5, 6, 7, 8 color-coded dimensions that the other methods can show you, you can explain that projection well. So it's kind of a very simple indicator to tell you, well, here we've got enough explanations, as I said before, you should probably be happy with them, or here you have a very high dimensionality of the data in this area of the projection, and I don't offer a tool to answer the explanation in that area, but it's just a warning signal indicating that you should not rely upon the few color-coded dimensions to understand the data in this area. Back to you.
Yeah, I also got that notion. Thanks for making that clear. I have received a question in the meantime from the audience, which was proposed by Chatai himself. He was asking about the borders between categories and if they have a meaning, or if they can be sort of special characterized, and you can make sense out of these borders.
Very good question. Short answer, yes. So if you look at the images you've seen before, they look kind of like shaded cushions, so stuff is bright in the middle of one of these colored regions and dark as you go close to a boundary. Basically, brightness tells you how certain the color-coded explanation is. So if you see something bright, it basically means that color that you color code there is a very good explanation for that local region. When things become dark, it means, well, the color that you code there starts being a less and less good explanation. So actually close to a border, you can think that both colors on both the left and the right side of the border, so to speak, should be joined in the explanation. Or both the left and right dimensionality shown in the color coding should be joined in the explanation. So the border is like, if you will, a logical end between the explanation on one side of the border and the explanation on the other side of the border. Back to you.
Thanks for that. Thanks, Alex, again. Thanks for that answer. And I think, having learned a lot about new explainers and the specifics of the wine data set regarding the alcohol and the sugar degree, thank you again. I will switch over now to the third approach, which combines three things, actually. It's about progressive visual analytics, it's about parameter space analysis, and about time series all together in one visual analytics approach. The presenter is Sebastian Bugge-Löschke. He is in the second year of computer science in the bachelor. Correct me if I'm wrong, Sebastian. At Aarhus University. And he is a student of Hans-Jörg Schulz there. And the title is Progressive Parameter Space Visualization for Task-Driven SAX Computation Configuration. Happy to hear your talk.
Hi everyone, I'm Sebastian Löschke from Aarhus University, and I'll present the short paper Progressive Parameter Space Visualization for Task-Driven SAX Configuration with the co-authors Marius Hochgräfer and Hans-Jörg Schulz. As time series datasets are growing in size, analyzing these datasets becomes more time-consuming due to the vast number of measurements collected and the large value ranges that individual data points can cover. To gain timely insights into such data, analysts often use data reduction methods to increase analytic throughput. Two widely used data reduction approaches are piecewise aggregate approximation (PAA) and symbolic aggregate approximation (SAX). PAA reduces the temporal resolution of the data by aggregating data segments by their mean, while SAX reduces the resolution by aggregating value ranges to symbolic values. SAX and PAA are usually used in concert by applying the SAX transformation on top of the PAA aggregated data. PAA takes a time series of arbitrary length and splits it into equal-sized segments of length omega. The mean value for each segment is then computed and used instead of the individual values for that segment. In this example, we have a dataset of size 54 and use a segment size of 3. PAA then computes the 18 segments that are displayed in the red figure. SAX discretizes the PAA aggregated data by mapping intervals of PAA segments to a discrete set of alphabet symbols. The PAA segments in the figure are mapped to the corresponding symbol that represents the breakpoint interval it lies within. Here we use an adaptive version of SAX that computes these interval boundaries using representative produced by Lloyd's algorithm. The result is a string of 18 symbolic letters. Thus, the output is reduced to 1/3 of the original data size, and with an alphabet size of 3, the value range is reduced to a 3-letter alphabet. While reducing the amount of processed data increases the performance of analysis algorithms, it at the same time decreases the accuracy of the results. Finding a pair of parameters for the right trade-off between the data reduction and the remaining utility of the data is a challenging problem. So far, finding the right set of parameters for PAA and SAX is either done in a user-driven way by asking the user to set the parameters based on their domain knowledge and expertise, or using an automated data-driven approach that relies on measures like descriptive statistics derived from the input data. However, none of these approaches take the analytic tasks to be performed on the reduced data into account. Tasks such as correlation or trend analysis might benefit from other parameterizations, which is not considered in these evaluations. We fill in this gap by proposing a semi-automated, task-driven parameterization for PAA and SAX through a parameter space visualization. The visualization shows the difference of progressively running a given analytic computation on samples of the raw data and on the reduced data for a representative set of data samples. Depending on the analytic task, a different computational analysis will be run, For example, for finding periodicity in light curves data, the greatest periodicity is computed, while for establishing trends, a regression analysis is carried out. Visualizing the resulting discrepancies for different parameter configurations allows the user to make an informed choice with respect to the gain from the data reduction and the loss from the error introduced for the particular task at hand. To cope with the bottleneck of computing all parameter combinations for the full dataset, we employ a progressive computation that generates early results of increasing quality and quantity. For the early results, the user can then steer the computation to compute interesting regions of the matrix as they emerge. We demonstrate this approach on two analysis tasks: finding periodic patterns in light intensity data to identify exoplanets, and analyzing trends in weather data to characterize local climates. Here we see a fully computed parameter space visualization. To determine the utility of the dataset after differently parameterized PA and sex transformations, we captured the analytic task by a computational function, which could be the function of finding the periodicity for a data sample. Computing this function on the data yields a numerical result, for example, the period of the light curve event. We can then compare the results by showing the absolute difference between the results computing on the original data and the results computed on the reduced data with the corresponding parameterization. Each column of the matrix represents the PA segment size, omega. Here we utilize the value range from 1 to 14, and each row represents the sex alphabet size alpha. Here we utilize the value range from 3 to 14. Each cell in the matrix also shows the error measure on a continuous color scale, where the smallest difference and thus the highest quality maps to the brightest color, and the biggest difference and thus the worst quality maps to the darkest color. Generating the matrix for all parameter combinations and for the full datasets requires an extensive computational effort that is not achievable in a reasonable time. By using a progressive approach, we do not wait for the whole matrix to be computed, but instead we visualize early partial results computed on subsets of the data that are then refined over time. From the early results, the user can then steer the computation to compute interesting regions of the matrix. Here we see the animation of progressively computing the parameter space visualization. The animation shows how different parameterizations are progressively computed and updated in the matrix to provide early results. Early on, we can see the regions of interest that emerge, and we can steer the progression towards computing these regions. Steering is done by selecting columns, rows, or individual cells of the matrix through clicking. Once cells have been selected, they are prioritized and fully computed before the non-selected cells. The visualization also includes progress bars for all columns and rows, and also a progression per cell. Hereby we indicate the degree to which the data has been processed across the particular set of parameter combinations. This serves as a visual guide for the user to determine both the quality of the partial results as well as the progress in the overall computation. In our implementation of Progressive Visual Analytics (PVA), we begin by selecting a representative sample of the full dataset. This selection is either done manually by the user or automated using appropriate algorithms. Then we divide the sample into equal-sized chunks and iteratively compute the error measure for each cell in the matrix. Here we use a Latin hypercube sampling to determine the order in which the cells are processed. This is to avoid oversampling individual columns and rows. After each full iteration over the cells, we continue with the first cell and the next data chunk. This produces partial results in the breadth-first manner. So instead of processing each cell in full, one after another, the computation can now be run per chunk with the ability to obtain partial results for each cell. As the figure shows, already after 1/3 of the computation has been run, we can see an emerging pattern. From this point, the user could choose to steer the computation to only run the columns for the segment size 1 to 5. Hereby, the selected columns would be computed before the rest of the matrix, and the user will be able to make an informed choice with respect to the gain from the data reduction and the loss from the error introduced. Now let's go into an example. For the application of our visualization to light curve data, we use publicly available dataset of light intensity emitted by stars. This dataset was measured by the Kepler Space Telescope during a NASA mission on discovering exoplanets between 2009 and 2013. To find extrasolar planets in these datasets, one is looking for periodic patterns. These are produced when a planet orbits around its central host star and periodically reduces the light intensity of the star during transit from the viewpoint of the telescope. To look for periodic reductions in light intensity, astronomers use the box-fitting least squares algorithm BLS. The figure here shows that the end result of the parameter space visualization for the 6 light curve measurements from the Kepler mission. Each cell in the matrix shows the difference of running BLS on the original data and on the reduced data for the corresponding parameterization. The parameterization of the data reduction shows that the error measure varies more strongly between neighboring columns than between neighboring rows. This shows that our computational task is not very sensitive towards the particular numeric values at each time point, but instead needs to maintain a high temporal resolution to capture the transit periods. One suitable pair of parameters is, for instance, an alphabet size of 10 and a segment size of 3. Using this parameterization for a computation of BLS on the first 10 quarters of the exoplanet 8b led to a computational speedup from 61.7 down to 20.6 minutes, which is a decrease in computation time by approximately 66%. In comparison, we also applied the matrix visualization to the context of weather data. Here our analytic task was to compute a linear regression model for the data. Here we used the difference in slope of the resulting regression line as an error measure for assessing parameter combinations for SACS and PAA. In the figure we see that the resolution of the value range is much more important, as small deviations in the values can have a large influence on the resulting slope. Therefore, the error measure varies more strongly between neighboring rows than between neighboring columns. By using a task-driven approach to determine a suitable parameterization of SACS and PAA, we do not look at the input data. Instead, we look at how the results of our analytic tasks on the reduced data deteriorates from the results on the raw data. By visualizing these results with a parameter space visualization, the analysis can make a choice on the trade-off between the computational speed gained from the data reduction and the error introduced from the data reduction. This result-oriented approach to the parameter estimation is made possible through PVA methods for computing partial results long before an exhaustive computation would be complete. Also, by being able to interactively steer the progression towards regions of interest in the parameter space. With the application of our task-driven approach, we were able to capture that some tasks are more sensitive to reductions of the temporal granularity, which we saw with the light curve data, while others depend more on the granularity of the value range. Thanks for your attention. You can find the tool on our department's GitHub page, and if you have any questions, feel free to ask me.
Thank you, Sebastian, for this presentation, and also congratulations, this very successful bachelor project, obviously. There is another shout out I would like to make for you, because you have this animation in your paper showing the progressive computation approach as an animation, which is very cool. I like it a lot. I want to tell you that. I have not received questions from the audience so far. That's good, because I have a lot of them. The first one that I have regards the error measure, or I would like to open with measures, because you are using several measures how to calculate this measure. And obviously you are sort of computing sort of back transformation from ZACs back to original time series, do some comparisons, and then you have maybe even downstream models like the regression. Can you tell us a bit more about these measures? And there is another question that I would like to add. Isn't there even another interesting analysis space coming up as different measures are available to do that?
The measure we have in each cell of the matrix is the error measure of computing this analytic task, which for the first example was the Boxley square algorithm applied to the light curve data. Then we first apply this algorithm to the raw data to compute the ground truth value. And then in each cell, we compare the SAX and PAA output with the ground truth value, and then show the error measure in a percentage difference from this ground truth error. Here you could also, it's also very important to show the trade-off between the error made and then the speed of and the data reduction gained by the algorithms. However, one can read from the axis labels how much data is being saved, and hereby get an impression of how much speed up in the computation, or how much speed up you would gain from this set of parameters. So here we assume that the analyst that has chosen to use SAX knows that by using a segment size of 2, you reduce the data size to half of the original data size, and for a segment size of 3 to 1/3, and etc. But here we chose to show the error measure in each cell of the matrix, since we said this was the most important thing for the analyst when he tries to find the parameterization.
No doubt, no doubt. Got that part. I just have a look. No questions from the audience so far. Then I would like to add one, which is sort of related to your answer, because you were already addressing the number of timestamps that you're using in the PAA. PAA is a very, very prominent algorithm to aggregate time, and then you are using this X descriptor to use the radio domain and discretize it. Have you considered to exchange PAA against other time series representations, or descriptors as they're also called? For example, linear piecewise segments or something like that, to open up this design space even more?
Yeah, here we tried with different approaches. Example, the linear piecewise or PIPS algorithm. But for the approach, we initially started with the approach to light curve data, and here we wanted equal size segments, which is what we get with PAA. Even though we've seen in other examples that PIPS increases the accuracy, or not the accuracy, but the overall slope or pattern of the time series better than PAA, but PAA keeps the size of the segments equal,
Constant,
which is why we chose to use PAA together with SAX.
I can understand that. And also that PIP will resemble the shapes better. Yeah, but sounds very interesting and promising, and also for future works. Congratulations once again. Just a last look. No further questions from the audience. Then I would like to thank you again, Sebastian, and I will come to the fourth paper of this session, which is presented of a group. I have to be a bit spontaneous now because the actual speaker is not showing up. There is a problem. He could not make it. We hope that this will change. Still, I will announce the paper and hope that they will appear. It's a group coming from the Texas Tech University, and the paper would have to be presented by Tommy Dang, who's an assistant professor in the Department of Computer Science. The paper name is called Cognostics, Visual Features for Doubly Time Series Plots. Here we go.
I'm going to present the paper, Cognostics, Visual Features for Doubly Time Series Plot. My talk has 4 parts. I will introduce the idea of the paper in the first part, and introduce a set of visual features in the second part. I then discuss some use cases of visual features for the time series in the third part, before giving some conclusions in the final part of the presentation. I'll move to the first part of this talk. As we know, scatterplots can display the 2D distribution of data so that we can gain some relation between pairs of variables in the data set. Scatterplots is a set of visual features that allow us to automatically extract scatterplots of interest. However, there is no temporal information in the scatterplot. In contrast, time series can describe the temporal values of individual variables, but cannot provide obvious association between variables. In this paper, we proposed the use of connected scatterplot that allows the strength of scatterplot and still retain the temporal information by color. We call this type of plot the doubly time series. So, what are visual features of the time series? To answer the question, I will move to the next part of the presentation. We report a set of 8 visual features for the time series, namely outline that indicates the existence of outliers, clumpy that indicate clusters, striated that measures the long thin lines in the plot, correlation that detects variables that have correlated changes, intersection that scores the number of crossings in the plot, circular that extracts loops in the plot, trend that points out the trend in the doubly time series, and finally the length in the mean length of doubly time series. Using outlier score, we can find any doubly time series that has abnormal data points in the distribution. Clusters, which are detected by clumpy score, indicate stable periods before a sudden change happens. The correlations in change of two time series can result in a near diagonal line distribution of the time series. The circular loops imply cycle patterns in two time series, and these cycles are usually offset. In other words, one variable reaches its peak before another one. Moving to use cases of the doubly time series. There are 3 use cases in this talk. Firstly, we found cluster in the doubly time series of the US employment data. This data is the number of employees in thousands of various economic sectors of all states in the US. The dataset is the collection of monthly time series from 1999 to 2019. Looking at the doubly time series on the left, the relative position of two clusters is from the top right to the bottom left. This position implies a sudden drop in both sectors, the accommodation and the food services, and the leisure and hospitality. The drop was due to the impact of hurricanes Irma and Maria in 2017. Moving to the doubly time series on the right, there are 3 clusters. The first one is on the top right corner of the plot. The second one is on the top left corner. And the last one is on the bottom left corner. The relative position between the first and the second cluster indicates a significant decrease in route producing, while accommodation and food services remain stable. The sector route producing was affected by the economic recession of the Virgin Islands. The moving from the second cluster to the third cluster happened at the same time as that of the left doubly time series. Also, the The relative position between the clusters points out the fact that wood producing was not affected by hurricanes. We also found the circular patterns in the U.S. employment dataset. The slide shows the time series of total private and financial activities of Massachusetts. The green loop is due to the Y2K recession, while the orange one depicts the Great Recession of 2008. The offset between orange peaks of two sectors indicate the fact that the Great Recession of 2008 affected financial companies before having impact on other private companies. The last use case is the outliers in correlation patterns of the time series of SP500 dataset. This dataset is from 1980 to 2018, and we focused on yearly time series of open and closed values of the SP500. Its time series has around 247 data points, no stock data on Saturday and Sunday. On the left doubly time series plot, we can see an outlier which is corresponding to the mini crisis on the 13th of October in 1989. The crisis was caused by the leverage buyout deal for UAL Corporation. On the right plot, the outlier is due to mini crisis on the 27th of October in 1997. We saw the impact of an economic crisis in Asia on the US at that time period. To sum up, the time series can display pairwise relations of variables in multivariate time series. We have proposed a set of visual features for the time series, and the use of proposed visual features have been discussed in the use cases. Thank you for your attention.
Unfortunately, we won't be able to reach the authors of the paper today. What you can, of course, do is put the questions on Discord, and the authors, I think, will then answer these questions later on. As it was about time, you can, of course, also ask Sylvia. Maybe she knows the answer as well. I would like to come to paper number 5, which is the last paper of the session. It's presented by Katarina Ruzzo herself. Almost a reason to introduce her today. She did this work together with Aida Nordman, also working at Linköping University. Katarina is a member of the community for a long time, coming from the visual data mining world. She had an emphasis on research on time and space, for example, together with Natalia and Janadi. And what she also did for a long time is to work with event sequences. And this brings us back to the paper she's presenting today. And I'm hearing that my technician was now able to come up with a video. As we had no questions, this was a bit more critical. And I can now stop talking and listen together with you the first presentation.
This work deals with the exploration of long-duration event sequences. Such sequences can be found in many domains, for example in healthcare, process control, and in eye tracking, which has been our focus. Eye tracking data are commonly collected with high frame rates and over long periods, and they are interesting in many monitoring tasks for understanding human behavior. Usually, when analyzing event sequences in search of common patterns, such patterns are sought between sequences, meaning that the combination of events is considered a pattern when it occurs in many sequences. But in domains such as case analysis through eye tracking, which deal with fewer sequences but with long durations, patterns are often sought within sequences, meaning that the combination of events is considered a pattern when it occurs repeatedly within a single sequence. This is the case in the domain we target in this work, namely air traffic control, or ATC, and specifically in air traffic controller training, or ATCO training. ATCO training is primarily performed using simulators, where ATCOs successively practice increasingly complex ATC scenarios while an instructor is observing them. Their performance is then followed up together with the instructor, and the choices they made while performing the scenarios are discussed. A current challenge faced today in ATC is that there are not many predefined scan patterns or formalized instructions explicitly stating how an ATCO is expected to scan their environment. So there is an increased interest for approaches concerned with how to formalize the process of training ATCOs and support instructors in the process of identifying and studying the visual scan patterns of trainees. One way to better understand the visual scan patterns of ATCOs in training is to collect eye tracking data while they are performing different ATC scenarios on the simulator and use it as support in the discussion between trainee and instructor. But analyzing this data and understanding what the trainee is looking at is a challenge for the instructor, because first, attention shifts rapidly and scanning tasks occur in seconds, so it can be difficult to detect them live. And second, as mentioned earlier, there are not many well-established formal scan patterns available that the instructor can look for in the data. Therefore, it becomes interesting to explicitly identify which specific regions the ATCO has looked at and in which order, and also to be able to make assessments about the correctness of these interactions, meaning whether the ATCO has looked at the correct regions in the correct order. Also, since the knowledge of what is correct lies with the instructor, analysis methods need to incorporate input from the domain expert. And this is where we make our contribution. So this is what we propose. Raw gaze data collected through eye tracking experiments can be converted into discrete event sequences by first dividing the field of view of the ATCO into distinct areas of interest and then aggregating the data into sequences of visits to these areas, forming this way the event sequences. Following this, sequence mining techniques could be applied in order to identify specific subsequences of AOI visits as patterns. However, since in this domain setting what is of primary interest is to detect individual scan patterns of different ATCOs, the mining process needs to be adjusted for seeking patterns within event sequences rather than between them. To do this, we have extended the exploratory pattern growth-based approach for pattern identification that we have presented in previous work to include a sliding window that splits the input sequence and a visualization that allows the exploration of the identified patterned events. So, in our proposed approach, introducing the use of a sliding window allows us to convert the problem of mining patterns within one long sequence into the problem of mining patterns in several shorter ones. Depending on the type of sequence we want to mine, the sliding window can be defined in terms of a duration or a number of events. More concretely, what we do is we use a time window which slides over the input sequence 1 unit of time in each step, resulting in a series of time windows, all of which have the same time span and 1 time unit difference in their respective start and end times. The size of the sliding window is set by the analyst, and the total number of windows that the sequence is split into can be computed with respect to the summed duration of the event sequences. So, if TE is the end time of the input sequence and the start time is 0, then the number of consecutive windows over the input sequence is given by the end time plus window size minus 1. The support of a sequence alpha is defined as the fraction of the total number of consecutive windows where alpha occurs. And alpha is a pattern if its support is above a given user-defined threshold sigma. So, for example, if we assume a sequence where events have no duration or equal duration of 1 time unit and define a window of size 4, we slide the window 1 event at a time and then mine the resulting sequences. We can then see that the sequence E followed by F appears in 5 windows and has a support of 5/12. While in an example of a temporal event sequence where events have different durations, we again slide the window of size 4 1 time unit at a time. And in this example again, the sequence E followed by F appears in 5 windows, but the support in this case is 5/16. An important point regarding the proposed work is that how the sliding time window size is chosen has a direct effect on the patterns that can be mined, since patterns longer than the time window can never be identified. This puts responsibility on the analyst to choose the size appropriately and highlights the importance of including domain knowledge in the process. The proposed approach has been implemented as part of the previously introduced prototype system called Eloquence, which has been extended with a new algorithm as well as an event overview representation. So the system is composed of three main views. The pattern tree view, used to interactively drive the sequence binding algorithm and display the patterns as they are being mined. The sequence view, which displays sequences as horizontal bars comprising the different events. Here, the user can choose to display the original sequences composing the dataset, or the split sequences on which the mining is being performed. And finally, the event overview is a representation of the distribution of pattern events across the event sequences. Timelines of each event of a selected pattern are stacked on top of each other to provide an overview of their frequency of occurrence and their distribution across the entire time period. All views of Eloquence are linked. Selecting a pattern in the pattern tree view will add the timelines of the events composing it to the event overview and highlight the pattern events in the sequence view. We have applied our approach to an eye tracking dataset composed of ATCOs performing landing and takeoff scenarios on a simulator. The data were collected using Adobe Pro glasses with a sampling frequency of 50 Hz. Following this, the continuous gaze data were translated to discrete sequences of AOIs visited by the ATCOs when performing the scenarios. To extract such sequences, a division of the field of view into 24 unique AOIs was created by an instructor. Here is an example of patterns identified from exploring a single landing scenario. The mining algorithm was run with a sliding window of 100 seconds, the maximum gap allowed between pattern events was set to 10 events, and the maximum time between pattern events was set to 10 seconds. The exploration of the data was initiated with a minimum support of 30%, and the pattern tree was expanded one level. Following this, the minimum support was reduced to 10%, and all branches were fully expanded. Exploring the pattern tree, we identify three central patterns. First, the initial approach pattern is composed of the AOI sequence runway radar, followed by weather observing system. Here, the ATCO is switching their attention between the radar monitor, where they are following the aircraft as it is approaching the airport, and the automated weather observation system, where they are monitoring the weather conditions. Highlighting the events composing the pattern in the event overview reveals that these are occurring primarily in the beginning of the landing scenario before the ATCO has made visual contact with the aircraft. The runway scan pattern composed of the AOI sequence out the window approach, followed by runway 1, followed by runway 2. Here the ATCO is repeatedly looking at the out the window approach area, anticipating visual contact with the aircraft, and then checking the runway for obstacles in order to give landing clearance. Inspecting the pattern in the event overview shows that runway scans appear more frequently after the ATCO has made visual contact with the aircraft and until the aircraft has landed. Finally, the landing roll pattern is composed of the AOI sequence out the window apron, followed by runway right, followed by runway center. The ATCO is inspecting the apron for obstacles and then follows the aircraft moving from the right part of the runway back towards the middle part, where the taxiway into the apron is placed. The event overview reveals that the pattern takes over after the aircraft has landed and repeats until it has entered the apron and stops moving. These three patterns reflect quite well the three main phases of the landing process and show the potential of the approach for identifying ATCO scan patterns. To conclude, with this work we have proposed an interactive sequence mining approach that allows an analyst to explore long-duration event sequences in search of patterns within them. The approach extends previous work on exploratory sequence mining by adding an algorithm that uses a sliding time window to split event sequences and then mines patterns from the time windows. Our objective has been to create an interactive tool that can assist an instructor in identifying ATCO's visual scan patterns while training on ATC scenarios. Such a tool has the potential to be used to assess whether tasks are performed as expected and form a base for discussion between instructor and trainee.
Thank you, Katarina, for this talk. I have not received any questions from the audience yet, so I will use the opportunity to ask you one of the questions that I've prepared for you when reading the paper. My first question is a methodological question. You were pointing out that you are using a approach that focusing on within sequences as opposed to between sequences, meaning that you focus on single one, as this one is long and you are interested in substructures. From a methodological point of view, what are your experiences with this sort of shift in the paradigm, and what can you tell us when doing this?
Well, more or less, from a pattern definition point of view, there is not such a difference, because oftentimes the patterns, the way they are defined, is like a combination of certain events that repeats, for example, often, if it's a frequent pattern one is after. So the difference here is that it needs to repeat often within one long sequence instead of between sequences. Now, from the application point of view, that has to do a lot with the sort of application area. And while commonly the sort of the most prominent approach is to actually look for patterns in large populations of data, in large sets of data, while in this case, this type of data has a particular character that is very long and a repetitive task. So, I mean, it's coupled to the definition of the pattern, but in a sense on how it is sort of implemented, I would say that from the methodological aspect, what differs is the implementation of the algorithm, not the sort of definition per se.
Yeah, that makes sense. That makes total sense. I've had another question, and still I would like to encourage the audience to also add questions. So far, one other question for me is sort of related also to your answer. There is one tiny limitation which you also discuss in the paper, and that's the length, the maximum length of the pattern that you can analyze. You say that, of course, you are involving domain experts in the particular case to ensure that even long patterns are to be grasped by your approach by defining the window length accordingly. Considering of guidance, could you imagine to combine it with an approach where simply subgrid sequences of any length would sort of be recommended if they are deemed interesting by some algorithm as a complement to user judgment?
That would be that's a very good point, and a very relative to sort of the theme of EuroVA this year. But it would be definitely interesting to have some sort of guidance in that way. And sort of the immediate thing that one could look at as future work would be how this size of the window, sort of at the simplest level, how the size of the window should be defined. So we could implement some algorithm that sort of pre-process perhaps the data and looks for sort of optimal window size to suggest. And that would definitely that would definitely be interesting to incorporate. Absolutely.
Talking about algorithms and measures, Shiva Magrawal is asking which measures, metrics apart from support you could use to judge the quality of a found pattern.
That's a very good question. I guess that at the moment we are looking sort of the quality of the pattern is very closely related to sort of the task and the user that is searching for it. So what we allow is we allow sort of the way a pattern can be defined would be in the way that we allow and apply our constraints, right? So depending on on sort of the preferences of the user and the preferences or the relevance to the task. One can be applying different sort of constraints and different definitions of what is an interesting pattern. Here we so adjusting sort of the length or the time difference between different events or the gap and so on are ways to define and alter, sort of specify the quality of the extracted patterns. And within this system also, even though we are measuring sort of the support in terms of frequency, however, it is not a global, it's not measured globally, but it's measured locally. So this mining approach that we are proposing concentrates on sort of local constraints and allows the user to adjust the frequency so that even infrequent patterns could be identified using this method.
Thank you for that. I have another question from Alex Talia directly, and he's asking, how would you relate this type of analysis to the more self-organized analysis based on eye-tracking bundling, which is also in this applied in the same area?
I'm not sure I understand the question.
Alex, you may simply unmute yourself and.
Yeah, sorry, thank you. Now, very briefly, so you do a, let's say, an analysis based on explicit mining of patterns, and then, of course, you present them visually, analyze them visually next. What I wanted to ask very simply is, in ATC or ADCO training, trail bundling has been used, or at least I have used it for a while in the past, to aggregate, well, similar eye trails and therefore find high-level patterns. But this is more in a self-organized manner that is without, explicitly having a pattern that you're looking for, distance between two patterns. So this is a more self-organized approach as opposed to what you present here. So I was wondering if you compared your proposal to the trail bundling, track bundling approach to see where are the differences, or if you could comment anything on that.
That's a very good question. Thank you, and sorry that I misunderstood. But that's a very interesting point, because the problem, so to say, is bigger than that. The problem with mining specific sequences in this manner is that the sequences can be very explicit, sort of. And the fact of the matter is that, especially in ADCO training, there can be like small variations that, even though, or especially in eye tracking data in general, that even though people may have the same sort of overall pattern, there are very small distinct dissimilarities that can be the cause of not identifying enough patterns. Maybe there are hidden patterns in a sense. So it is very interesting to look at sort of similar patterns and bundle them together. And this is something actually that we have now in the pipeline for future work, because we haven't explicitly sort of addressed that yet. But how to handle this type of sort of small variations in the patterns that actually don't mean that they're a different pattern completely, but actually are sort of implying the same behavior between them. So, short answer, we haven't addressed that yet, but it is a very relevant and in the pipeline to look into adjusting sort of our algorithm for handling this type of variations, and bundling the tracks in that manner is a very good suggestion. Sort of what one could look into how different patterns could be grouped into sort of a meta pattern, so and mine in different levels of detail. So very interesting point and very correct.
Clear. Thank you. Thank you.
I would like to add one last question, maybe a short answer. Always when you design a sliding window approach, the designer has to make this trade-off between the duration of it, so the temporal domain, or the number of indices used. In this particular use case, had you problems with defining these parameters?
Yes, problems and problems. In this particular use case, we have found that, in a sense, it makes more sense to define the window in terms of time, since you can sort of the experts can point that, okay, there are certain time windows or time periods in which things change, sort of, or in how the articles perform their tasks. So it's very difficult to implement it in terms of number of events, mainly due to the sort of nature of the data and due to the nature of the task. And when I talk about the data, it's because there are we have used the eye tracking glasses to collect data. So as soon as someone moves their head around, you can have events in terms of eyes not found, or they are looking outside sort of the field of view that is of interest. So you have events that are undefined, which makes it more difficult to actually use the number of sort of events, since it's more unpredictable than the actual length of a window in terms of time.
Makes totally sense, as this data is particularly not quantified.
So of course one could pre-process their data, and if you would sort of take off these sort of more uninteresting events, perhaps it would make more sense. And also in another application area that is less sort of attention is less shifting so much, then it could be more appropriate to use a number of events as
makes sense to me. With this, I would like to thank you, Katarina, in specific, and let's thank all 5 author groups again for proposing this work and presenting it today to us. Thank you from my side also. And I see claps in the chat. I think the people have liked it. Thanks again for having me as a host. I would like to ask Katarina if she wants to take over and add any additional remarks for the program. And from my side, have a good day. And once again, greetings from Vancouver, where it's bright already.
Thank you very much, Jürgen, for hosting this session. And we finish even on time, which is unusual for the day. But now we will have a short coffee break. The time is now 3 in Sweden, and our next session will start at 3:30 with another set of exciting talks. So thank you, everyone. Thank you again, all the speakers, and thank you, session chair. And we see you all in the next session in half an hour. Thank you very much.